MCAT 2017 Cram, Biochemical and Biological Foundations of Living Systems. Passage 22, the speed limit of DNA polymerase. As you view the reading of the passage, you'll notice some highlighted snippets of text. What I want you to do is garner meaning from these specific selections in order to answer the questions that follow. Good luck and happy reading. Paragraph 1. During DNA replication, a double-stranded DNA molecule is split apart into two single strands. Each of these strands acts as a template. So here goes one of the original template strands. A new complementary strand is generated along with it. This process depends on DNA polymerase, a molecule or enzyme rather, that travels along each template strand and catalyzes the formation of a new strand. Okay? DNA polymerase adds adds bases that pair to the bases already present in the template strand, forming base pairs that are rungs of the DNA molecule. The average distance between consecutive base pairs in a DNA strand is 3.4 angstroms. Note that 1 angstrom is equivalent to 10 to the negative 10 meters. DNA polymerase can replicate a DNA strand even as it follows the twists and bends of the DNA strand. Okay, so here we see the DNA strand is neatly unwound, but it can even do this along twisted coil DNA strands. Okay, all right. Figure 1. DNA polymerase replicates template strand. So there's replication of a new strand that's anti-parallel to the template strand. All right. Paragraph 2. The DNA polymerase molecule can form new base pairs quickly. However, quantifying its maximum replication rate is difficult because individual polymerase molecules can accelerate, decelerate, pause, or fall off the template strand. Polymerase replication rates are often reported in base pairs times seconds to the negative 1 power or base pairs per second. The number of base pairs generated per second. All right. Paragraph 3. One experiment determined the maximum replication rate of single DNA polymerase molecules by measuring the elasticity of single strands of DNA as they were being replicated. The total number of bases in the DNA molecule, denoted N subtotal, was known. The elasticity of the molecule at each point in time was compared to the known elasticities of single-stranded and double-stranded DNA of the same length in order to infer the percentage of the DNA molecule that had been replicated, which is denoted P of T. Finally, the number of base pairs replicated at any given time, N of T, was calculated using the following equation. N of T is equivalent to P of T times N subtotal or N total. Results of this analysis are shown in Figure 2. So let's take a look at Figure 2. Figure 1. DNA polymerase replicates template strand. So here we're shown the activity of DNA polymerase. On the abscissa, the x-axis or the input value, we have time in seconds. And on the ordinate or the output value, we have the number of base pairs replicated as a function of time, okay, which is denoted N of T. So here we see there's a rise as time progresses in the number of base pairs replicated. All right, okay. Which of the following postulates may explain the plateaus in Figure 2? Okay, so these are the plateaus the question maker is discussing. Is it going to be A, single-stranded DNA can form hairpins that force the polymerase off the template strand? Is it going to be B, DNA contains specific sequences that cause the polymerase to pause? Is it going to be C, DNA structure is highly irregular, allowing the polymerase to move at a constant speed? Or is it going to be D, the replication rate of DNA polymerase depends on the surrounding concentration of bases? This is a tough one, so definitely press pause if you need to. You may need a tad bit of outside knowledge of genetics to get this one, or the biochemistry subtopics of genetics. All right. Okay, so let's see what we have here. The question is asking you to focus on the plateaus. So this region and this region, okay, the flat areas of the graph. During the plateaus, time is basically passing, but DNA polymerase is not adding any new bases to the template strand, okay? Therefore, the polymerase has obviously paused during the plateaus. That's just a little background for anyone who may not be familiar with what a plateau signifies in a rate of time graph, okay? Thus, the correct answer is that DNA contains specific sequences that cause the polymerase to pause, because if it's not moving, it's paused, okay? All right. At approximately what time does the DNA polymerase molecule in Figure 2 experience the greatest positive acceleration in rate of base pair generation? Is it A, 375 seconds, B, 425 seconds, C, 550 seconds, or D, 75 seconds? I'll give you a moment to decide. This is a little tricky, so try to read the graph carefully, okay? All right. Okay, so acceleration is normally defined as the change in velocity, speed, which is speed and direction, okay? But since we're kind of ignoring the change in direction, although DNA has twists and bends in it, we're looking for the greatest positive acceleration in terms of increase in speed only, okay? This graph is quite similar to a distance versus time graph, except that the y-axis, n of t, indicates distance in terms of how many base pairs the polymerase has traveled along the DNA strand, okay? Therefore, the slope of the graph will tell us the speed of the polymerase in base pairs per second. So if we go to 75 seconds, which is halfway between 50 and 100, we see that the slope actually decelerates. It goes from increasing to flatline, okay? So answer choice D is out at 425 seconds. And 550 seconds, it's kind of the same thing. At 425, we experience what we experience at 75, and at 550, similar experience, okay? However, let's see, at 375, which is what we have left, we see like an increase. Thus, we can confidently say that at 375 seconds, the slope has the greatest positive acceleration and rate of base pair generation out of these particular selections, okay? All right. So if a single-stranded DNA template is 6.8 micrometers in length and a single DNA polymerase enzyme requires approximately 4,000 seconds to replicate it, what is the average replication rate of the polymerase enzyme? Is it going to be A, 5 base pairs per second, B, 1.4 times 10 to the third base pairs per second, C, 5.0 times 10 to the third base pairs per second, or D, 1.4 base pairs per second? You decide. Definitely press pause if you need to and refer back to the passage if you need to. You may have to use some basic algebra skills, unless you're a genius, which I'm sure a lot of you are, and you can just do this all in your head. Either way, good luck. All right, let's break this down. Okay, so from the passage, we know that replication rate is normally expressed in BP per S, or base pairs per second. And to find this, we need to determine two things: the number of base pairs in 6.8 micrometers, and two, the time it took to do this, okay? Well, we kind of know the time, so we just need to make the fraction per second. So, okay, again, the length of the DNA strand in question is 6.8 micrometers, and we know from scientific notation that micrometer is going to be mu m, or 10 to the negative 6 meters, okay? And from this, we can determine the number of base pairs because the passage also tells us, quote, the average distance between consecutive base pairs in a DNA strand is 3.4 angstroms, where angstroms denote 10 to the negative 10 meters, okay? So here goes the math for converting the number of base pairs and the time given into a rate. So we convert 6.8 micrometers, or mu m, into 6.8 times 10 to the negative 6 meters over 1, because we're going to do some dimensional analysis here. Now we're going to multiply that by the second piece of information I just mentioned that's also given in the passage, 1 base pair per 3.4 angstroms, or 3.4 times 10 to the negative 10 meters. Let me write in the meter thing. Okay. Alrighty then, so the next thing that we have is to solve this, to simplify it. So when we take out the meters, we're going to get 2 times 10 to the 4th base pairs, because remember, whenever we divide exponents, you're basically just excuse me, you're basically subtracting them. So negative 6 minus negative 10 is the same thing as adding 10 to negative 6. That's going to yield a positive 4 exponent, okay? So now we want to simplify the number of base pairs per second. We want to get a timed rate, okay? And so when you divide 2 times 10 to the 4th base pairs by 4 times 10 to the 3rd seconds, which is the same thing as 4,000 seconds, you're going to end up with 5 base pairs per second. So the correct answer choice is answer choice A. All right? Okay. A scientist allows a single-stranded DNA molecule of known length to undergo replication with a DNA polymerase for a known time interval. At the end of this interval, she records the elasticity of the DNA molecule. Based on the passage, which quantity can be calculated? Is it A, the total number of pauses in base pair generation, B, instantaneous velocity of base pair generation, or C, average speed of base pair generation? Definitely press pause if you need to, and I'll give you a moment to think. You may want to refer back to the passage. All right. Okay, so this is basically the equation of interest from the passage that we're going to use to arrive at our final answer. Okay, so we have measurements of time and elasticity. And based on the passage, the elasticity, denoted P of T, can be used to, quote, infer the percentage of DNA molecule that has been replicated. So since the DNA is of known length, okay, N of T, we know this, this means that scientists know the number of base pairs and total, and therefore the distance traveled by the polymerase, okay? If you also refer to figure 2, the graph. So based on the time interval and the distance traveled, we can calculate the average speed or average velocity, but we're going to eliminate instantaneous velocity. And total number of pauses, there's no mention of that. And we can't infer that based on the information that we're given. Okay, so velocity includes direction, and since the DNA molecule can twist and turn in a variety of directions, we can't predict the velocity based on the given information. Therefore, we can only calculate the average speed of DNA polymerase. Okay, so that's our final answer, average speed of base pair generation. Which feature of figure 2 represents the maximum replication rate of a single DNA polymerase molecule? Is it A, the highest slope value, B, the highest N of T value, C, the highest T value, or D, the highest plateau length, or longest plateau length rather? You decide. I'll give you a moment to think. Definitely press pause if you need to. All right. Again, let's break this down once more. So the replication rate is the number of base pairs per second, or base pairs times second to the -1 power, however you want to think about it. Okay. The y-axis in figure 2 is N of T, and this is basically the number of base pairs synthesized. And the x-axis, T, is the number of seconds that have passed. To find the number of base pairs per second, you basically would divide the number of base pairs that had been replicated, N of T, by the number of seconds that have passed. So, in other words, the rise divided by the run. Okay, this is an expression that some people are familiar with. Not all teachers throughout one's lifetime have referred to the values on the abscissa as run and the values on the ordinate as rise. Okay, some people are familiar with this. So if you've never heard of it, now you heard of it. In other words, the replication rate of DNA polymerase can be found by dividing the rise of the graph by the run of the graph, okay? So rise over run in simple basic algebra is the definition of the slope, okay? Therefore, the slope represents the replication rate of DNA polymerase. And so the highest slope value will be the maximum replication rate of DNA polymerase. All right. That's it. It's over.